Good evening. As you heard, my name is Jim Graham, and I head up Keynote Therapeutics. We are developing a new class of cancer drugs based on technology that originated actually here in Fort Worth at UNT Health Sciences Center and at NIH. And it's my great pleasure tonight to introduce to you Dr. Greg Aune. Dr. Aune is a professor of pediatrics at the University of Texas Health Science Center in San Antonio, where he holds the Greehey Chair for Cancer Survivorship, as well as the Stephanie Edlin Chair for Pediatric Cancer Research. Absolutely. Greg began his journey with cancer at age 16 when he was diagnosed with Hodgkin's lymphoma. He survived cancer but did not escape the long-term after effects of his cancer care. He had open heart surgery at age 35 to address damage caused by the cardiotoxic cancer drugs that he took, and to this day deals with the long-term health implications of cancer survivorship. But his battle with cancer did not limit Greg. Instead, it drove him. It drove him to dedicate his career to caring for children with cancer as a pediatric oncologist, as a research scientist, and as an advocate for childhood cancer awareness. Dr. Aune trained at leading centers such as MD Anderson and Johns Hopkins. He now leads his own lab in San Antonio, supported by the NCI, St. Baldrick's, Hyundai, the Pablo Foundation, and relevant to all of us here tonight, the Rutledge Cancer Foundation. Greg is the kind of person who sees patients all day in the clinic, runs over to the lab to check on the latest set of experiments, goes home and helps his 4 children tend to the pigs they're raising for 4-H, and still has time to talk to me about the next step in our drug development process. It's an honor to have Greg with us tonight, so please join me in giving him a warm welcome to the stage.
So they tell me my mic is live, so that's good. So Jim told you, thank you for that nice introduction, Jim. That was too kind. Jim told you a little bit about my history. I'm going to start off tonight with a little more detail on what he just discussed. The overall title that I'm going to talk about tonight is New Drug Development Strategies for Childhood and Adolescent Cancers. I got to find the clicker that they gave me. Forgot I had that. We
love you, Greg.
Thank you. All right, so I'm going to start talking about my experience as a Hodgkin's patient. So I was diagnosed with Hodgkin's lymphoma in the day after Christmas in 1989. I think most patients remember very vividly the day they were diagnosed. So my story is about 6 to 8 months before that day, I'd had a lump above my right clavicle, and I'd been to the doctor at least 3 times. Did not probably get the right workup, but ultimately, the Christmas Eve, it swelled way up. It got about as big as my fist. So my mother took me to her internist, an adult doctor, the next day. And when we were there, he took a chest x-ray, and I remember him coming into the room, and he said, "Here's a normal chest x-ray." And then I'm like, uh-oh, what's coming next? And then he put my chest x-ray up there. And even then, as a 16-year-old, I knew there was something very wrong with that chest x-ray, and it's because I had a big tumor in my chest. And I was diagnosed with Hodgkin's lymphoma that day, and it was probably one of the biggest life-changing events that I've had. It turned my life upside down. It was a really difficult experience to get through. I can say 30 years later, there probably has never been a day that I've been on this earth that I haven't thought about it. I still think about it. But I was fortunate. I survived. And what I took away from that experience was mostly good. I think I learned a lot. It has defined my life. But what really drove me was the experiences I had as a cancer patient. And so I wanted to go through my treatment. I think many here are familiar with cancer treatment. So in 1989, we treated Hodgkin's disease a little differently than we do now. So I had a splenectomy, which we do not do now. I had about 9 weeks of radiation, some to my chest and then to my abdomen and pelvis. The hope then was that that would eliminate the disease, but unfortunately I only had about a 40 to 50% reduction in my tumor size. So after that I got to enjoy chemotherapy. And for that I got 8 cycles of chemotherapy with 8 different drugs. So I got a regimen that we don't use anymore, because what we've determined about this regimen is that it didn't really give better cure rate, but it caused a lot more toxicities, both acutely and long-term, it turns out. But it worked, so I survived. In my era, there wasn't, I don't know if any of you are familiar with a drug called Zofran, but it's a very great anti-nausea drug. Zofran got approved by the FDA, I think 2 weeks after I finished my chemotherapy. So unfortunately, I didn't get to take that. I used to throw up sometimes 24 to 48 hours straight with my chemotherapy. I got something called anticipatory nausea, where I would throw up the morning of my treatment before they ever gave me anything, which really kind of messed with my mind. It's like, that's very scary, like that it can affect me that way. But I think this experience taught me a lot. I saw many children around me that were my peers in treatment that I would come to clinic and then they wouldn't be there the next day. And you'd say, what happened to so-and-so? And they'd tell you, and they had passed away. So I always told myself, if I get through this, I'm going to try to do something about this toxicity, because I'm one of the lucky ones. And I took with that a sense of responsibility that I had to do something with my good fortune of surviving this. So what I did not know at the time was the late effects. And so when I went through cancer, I think it was posed to me as, just get through the treatments. When you get done with them, your life's going to go completely back to normal. And that is not the case. I think cancer I tell my patients, cancer is a lifetime diagnosis. If you're diagnosed with cancer, you're going to expect to go to an oncologist at least once a year, eventually for the rest of your life, because of what I've experienced. I think I've experienced this as our field has learned more about survivorship. So what came for me next was I went off to college and medical school, and I was really motivated to try to get into medicine and then ultimately do research was something, that interest that came later. But I was relatively healthy. I had, I think, a lot of, kind of post-traumatic stress, just having to deal with, what had happened and why had that happened, and just trying to use it to my advantage. But then I started to have some health medical conditions. So in my first year of medical school, I was diagnosed with hypothyroidism, which is easily fixed. This was actually one thing that we knew was coming, because I'd had radiation to my neck and they told me that was likely. So I started on some medicine for that, and that was great. I've had pulmonary fibrosis, which in layman's terms is scarring of the lungs. It's gotten progressively worse my entire 30 years as a survivor. What that means from a clinical standpoint is I've had 2-week admissions to the ICU for, I would say, mild or moderate viral infections that I just couldn't clear and ended up in the ICU. Because of that, I was really careful during COVID and was so happy when the vaccine came out, because I was high risk. I didn't actually see patients for that year until the vaccine came out. And fortunately, I have great colleagues that were able to take over and fill my service time when I was supposed to be on. I suffer from infertility. So one of the downsides of that regimen I took for chemotherapy was that I think in the studies on those patients, it was 100% rate of infertility. But fortunately, my mother had read an article in Reader's Digest when I was a patient, absolutely, about infertility. And she asked my doctor if they could take me over to, grew up in Eastern Washington, could take me over to Eastern Washington, or to Seattle, to donate sperm before they started my chemotherapy, and he allowed that. And so I gave some samples. It's the most embarrassing thing of my life as a 16-year-old. They told me at the time this would never work right now, but hopefully the technology will improve, and by the time you'd need it. So I have 2 sets of twins. They're now, thank you. They're now 13 and 18. And they're from IVF, from the sperm samples I gave that were unthawed over 18 years later, which is pretty incredible when you think about it. When I was a first-year oncology fellow in San Antonio, I had 4 kids. My second set of twins were 6 months old, or actually 3 months old when we moved to San Antonio, and I was super tired. I couldn't walk probably from here to that balloon without being really short of breath, and I knew there was probably something wrong. I was exhausted, but I kind of thought that was from having done a lot of inpatient months at the end of my residency and then having new kids. I was just super busy. But it turned out my instincts about a heart problem were right, because I went into the doctor when we finally got settled, and I had critical aortic stenosis, and during the workup for that and planning for surgery, I also found that I had 3 coronary arteries that were 95% blocked, and I underwent open heart surgery a week later to replace my aortic valve and do 3 bypasses. That was another major life-changing event. I don't think I handled it, honestly, as well as I did the Hodgkin's disease at age 16. I think as an adult, I was much more whiny about it, and it took my wife to just tell me, you need to, buck up and get things done because you got a lot to do yet. And so sometimes you need people in your life like that to tell you, and I, that was a big wake-up call for me. It really focused my research and my clinical career because of what I experienced then. Unfortunately, it doesn't end there. I've had multiple skin cancers removed off my back in the radiation field. I had a stroke in 2014, a mild stroke, probably one of the scariest things I've ever experienced. It lasted about 15 minutes of not being able to move the left side of my body. Fortunately, it resolved on its own. I ended up, when I went to the hospital, there was no permanent damage, and I haven't had issues since. So feel very fortunate for that. During that admission, I'd actually been trying to be more healthy. I think had been working out, trying to eat better, which I don't have a good diet. And I think I'd lost 10 pounds. During that admission, they tell me, oh, you have diabetes. So the radiation to the abdomen is a big risk factor, we now know, for the development of type 2 diabetes in our patients, and that's what happened to me. It's a big contributor. And so that was super fun. I was not happy about that, but you can deal with diabetes. And then now I deal with a second valve in my heart that is failing and potentially is going to have to be replaced in the next 18 to 24 months, hopefully longer. So if I sound short of breath, that's why sometimes when I talk, I get kind of short of breath. But I'm okay, I promise you. I take 21 pills a day. I tell my patients this that I see in clinic, because sometimes they complain about having to take 1 blood pressure medicine. I'm like, guys, I take a handful of pills. If you want to have good health, this is a preventive strategy. And so I always start with my story, because I think my story, it's pretty extensive in terms of late effects. I don't tell you this because I want you to feel sorry for me. I just want everybody to understand what happens to survivors, because the reality is 100% of survivors are going to have some type of late health effect that's the direct result of the treatments that we give them. And so I think we have to understand that in the field of oncology, because this is a new era and we need better treatments, and that's why I got into the research that I do. And so this is a picture of my family. This is a couple of years old. My kids are bigger than that now. But that's Noah and Emma are my older twins, and Elijah and Sophia are my younger twins, and that's my wife, Christy, who's a neonatologist and my biggest advocate for sure. So my personal missions in my academic career are 3. Clinical care. So I really focus on long-term survivorship care in San Antonio, trying to develop a comprehensive clinic for our patients, and then also work on developing with our adult medicine colleagues in the cardiology division a specific program to deal with cardiotoxicity, which is a big problem in the survivorship population. I'm an MD PhD, as Jim noted in his introduction. And so I do bench research and I see patients. Most of my time is bench research because that's what it takes to be successful, is a lot of time. And So I focus on research that tries to understand how chemotherapy, radiation, and other medicines damage the heart and other organs and cause late effects. And so for that, we use mouse models. And then I think another critical aspect of that work is trying to develop novel agents that essentially have a wider, what we call therapeutic index, meaning they will go to the tumor and not affect another organ. And so what I'm going to show you tonight is some of the work that the Rutledge Cancer Foundation has been instrumental in funding and through collaboration with Keynote Therapeutics. The Rutledge Foundation introduced me to Jim and his company, and I think that's been a wonderful relationship. And then my third main area is advocacy. So I got into advocacy early in my faculty career because I realized my personal story resonates with people, and I have some scientific knowledge and medical knowledge, and so I can hopefully tell the story, create awareness, help leaders understand what are we up against in our field. So I said that we're in a new era of oncology, and I truly believe that. So in the 21st century, 5-year survival rates for all newly diagnosed patients is greater than 75%. So if you go back to the 1930s and 40s, survival from childhood cancer was 10% at best. So some of my attendings, physicians at Johns Hopkins that were near the end of their careers, told me a story one time about how they would take the kids with leukemia and they would put them in another part of the hospital and they would let them die. And they would just provide comfort care. So we're not in that era anymore. We have good outcomes. Yes. Unfortunately, even with all our progress, cancer is still the #1 cause of death by disease for children in the United States. Across the world, 500,000 children will get cancer. And Western medicine has not arrived in many places. So survival across the world is not like it is in Western countries. So we have a lot of work to do there. And then I hope you, at least from my part, my survivorship story, understand the significance of toxicity and late toxicities. That's a huge part of the future for us. And in 2020, we have about 500,000 survivors of pediatric cancer in the United States. This is growing. One thing I think is critical for the Rutledge Cancer Foundation's mission is adolescent and young adults. So they have the same type of issues. Just because you're 20 and got treated for cancer doesn't mean that you're not going to have late effects. They have many of the same issues, and it's actually even bigger number of patients than the adolescent and young adult population. So what are our remaining obstacles? I think about them as two. We still have 20% of patients for which we don't have good treatments. Patients die. And even there are diseases that we consider that we're very successful at, something like Wilms tumor, for example, which has favorable risk. Wilms tumor has a survival rate of 90%, and there really isn't that much research in it anymore. But I think about it, would you want to pull 1 out of 10 numbers that said you lived or died? Because that's really what it means. 1 child out of 10 is going to die from that disease. So we still have far too many children that die. And then the second part is the long-term toxicity. So we need to approach these problems in a different way than we have in the past, because it's the situation has changed. So how are we going to do that? So first is I want to give kind of a brief, simplified version of how drug development has historically worked. So the first is, for cancer drug development, what historically has been done is we establish cell lines from patients' tumors. We take a tumor, we grow it in the lab, and then we try to understand what happens with that cell line to cause a cancer, and then identify potential ways in which we could give a drug and kill that tumor cell. Then if we find something, then that's moved into animal models, where we give a mouse a tumor or develop a genetic model of a mouse that the mouse spontaneously develops a certain type of tumor. And then we test those drugs. Then we go into clinical trials, phase 1, 2, 3. This process takes a long time. And for pediatric and adolescent young adult cancers, Drug development has lagged far behind adults. We just are not, it's not that fast. The other issue is late effects. So in this system, what we've done historically is patients get standard of care treatment. We wait decades, like where I'm at, 30 years off my treatment. And then we have quantified all their health problems and then made correlations between the exposures, what kinds of health problems they got, and then try to, say this treatment causes a certain late effect. But the problem with that is you have no idea mechanism because you didn't study the patients when they got exposed to these drugs. So again, it takes a fundamentally different approach to solve this problem in our drug development approach. So I got introduced to Jim and Keynote Therapeutics because they have a novel technology drug delivery platform, and it's called MIR5A. I'm going to try to not get too technical here, but I'm just going to explain what this is. So it's something that's called the nanoparticle. And a nanoparticle is the size, oh, hang on, I thought I had a pointer, is the size of nanometer size, so that's a measurement. It's small, 10 to 20 nanometers. For comparison, a red blood cell is 8 micrometers, so this particle is about 1,000 times smaller than a red blood cell. It's comprised of a peptide on the surface, which targets the drug to a specific receptor called SRB1. It has something called the myristic acid tail, which is hydrophobic, meaning it doesn't like to be around water, which is why it forms a ball like this. And then inside the particle, you can put drug payloads. And so for our work, you can load anything that, theoretically that is really hydrophobic, and Jim's company has tried to focus on chemical entities that look like they will assemble. And the group at North Texas that developed this had done a lot of work in trying to, determine which are the best compounds to put in there. We had them put anthracyclines in there. Anthracyclines, doxorubicin is one of the critical medicines we use in pediatric oncology. About 50% of patients will get an anthracycline. But those are the drugs that cause cardiotoxicity, heart problems in about 20% of patients at least. So we really need drugs that have that kind of efficacy, but don't have the cardiotoxicity problem with them. So we put novel anthracyclines in the particle, and then the idea is that it would go to the tumor. What's additionally attractive about this strategy is that the target, SRB1, this is a picture of publicly available sequence data. So essentially what this looks at is expression of the target receptor on a whole host of normal human tissues and the level of what's there. And so importantly, I point this out because there's tissues that have high expression, such as the adrenal gland, the liver, but the heart is clear over here on the right with low expression. So that's kind of what you want. You want something where there's a target that potentially has importance in tumor growth and development and is on a tumor, but you want it to have low expression on key tissues. And with anthracyclines, we really have to be careful and focused on causing heart issues because that's the big problem with them. So the target, importantly, we've found is expressed on subsets of Ewing's tumors, which I know many in this room are familiar with. But this is something called the Kaplan-Meier survival curve, and this was taken off patients' tumors were removed, their genetic background was sequenced, and then you can get this data that's publicly available. We sorted it based on the expression of SRB1 and looked at survival, so the target. So the target itself, if the target is high, the blue, survival, you can see, is dramatically different. It's at about 60 months, 100% of the patients in that group had died. Whereas if the target's low, there's many patients that are still alive, 60%. So the target seems to have, from this small subset of samples, some predictive ability with survival. So it's a good target because if it's highly expressed, those patients might be very amenable to treatment with this strategy. So does it cause cardiotoxicity? So what we've done in the lab is we've taken the particle with different anthracyclines loaded in it, and then we've given it to mice along with what I would term the gold standard for cardiotoxicity induction. So doxorubicin, the DOX, that is a drug that I mentioned previously. It definitely causes cardiotoxicity in the context of this experiment is what I would characterize as a positive control, meaning I know it's going to cause cardiotoxicity. And then we compared it to other particles that had either empty particle, nothing in there as a payload, or two different novel anthracyclines, 8198 and MIR5A. And so if you look at something called ejection fraction, which is a measurement we use in the lab to, it's an ultrasound, it measures how well the heart functions. And so with doxorubicin, at baseline, before we gave the animals anything, all of their ejection fraction is above 70%, if you look at that data. On the right-hand side, the dox group had about a 10 to 15% reduction in their cardiac function. The entities that had either just the particle or anthracycline in the particle, they have no effect. And we've done more of these experiments, and we have really good data that shows when you put these anthracyclines in the particle, the animals don't get nearly as sick, they gain weight, they grow as we would expect, and they don't have any cardiotoxicity. So we think this is a really interesting strategy potentially to further develop. We've also begun to look at this strategy in tumor cell lines. So this is something we call a dose response curve, and we are using a Ewing sarcoma line in a petri dish, and we take that cell line and we create different concentrations of the drug. So along the bottom is the different concentration of the drug, and then on the y-axis, the one going up and down, that is a measure of how much tumor kill you get. And so these curves are comparing 8198, just not in the particle. So I call it unencapsulated. I sometimes call it naked 8198 when I'm talking to my lab. But that gives we often talk about what's called the IC50. This is the dose of drug that you use to cause a 50% reduction in cell growth. And it's a way you can compare different conditions. And so for this, with the unencapsulated, we get a IC50 of 1.3 micromolar, which is a pharmacologically relevant dose. That dose should work in patients. When you put it in the particle, you actually get an IC50 of 1 micromolar, which is a statistically significant difference, and it's a lot lower. And that's because these cells have SRB1 on their surface, and we think that when it's in a particle, the particle can engage with the receptor and deliver drug more effectively. We weren't actually even anticipating and seeing this result. We were hoping just to show they were equivalent. Where it's really going to come into play is when we put this into a model where a mouse is bearing this tumor and then we treat it. Because then the medicine will circulate and go right to the tumor. And that's where we think we're really going to see some big differences. So future studies. This is just a small amount of what we've done in the lab. I can tell you we're really hoping that by spring we'll potentially have this ready to go for publication and move on to the next steps. So we have a lot more work to do, but we're very encouraged by these early findings. So we need to do that. We need to do what I would call in-mouse studies. So give a mouse a tumor and then give them our different treatments and show that the encapsulated anthracycline effectively kills the tumor. And what's unique about our effort is we're simultaneously going to look and show that it doesn't cause heart damage. And I know we can do that. The second thing we're beginning to think about is how does this target What types of other tumors is it expressed on? Sarcomas, other adolescent and young adult cancers, because it really hasn't been studied that well. It's a fairly newly recognized target. And we want to understand more about this idea that tumors that have high expression are more aggressive and more propensity to metastasize. So that's on our future studies list. And then we want to know what other cancers have this target, because we want to broaden the applicability of this strategy. And then if we can do all that, the great hope is that we will get to a clinical trial. I would not give a timeline on that, but the sooner the better. And we all know that in the room.
2 years.
Laura says 2 years. We'll do our best. So I wanted to end with a quote and then some acknowledgments. So this is Albert Einstein. Everybody knows who he is. He says, problems cannot be solved with the same mindset that created them. So I look at oncology, the pediatric oncology, adolescent young adult oncology. I look at it as a new problem. We've caused a new problem by being so good at going from 10% survival to 80% survival. If we're going to move the field forward, we have to think about these problems differently. And honestly, that's the future of our field. So to end with some acknowledgments, someone asked me at our table how I got involved, and I'll tell the story. So we were up at Austin, at the Texas Capitol. I think it was 2017 we were talking about this. We were there. One of our patients from San Antonio's mother's a judge in the Corpus Christi area, a Ewing sarcoma patient. Yeah, she's a Paralympian, and she was just in the Olympics in Tokyo. Yeah, she's pretty amazing. So they had organized a lobby day at the Texas Capitol where the Senate and the House read a proclamation about childhood cancer and declared a childhood cancer a day. And so I was there with a group from San Antonio, and I met Laura. And I remember she told me, what do you do? And we started talking, and she says, I really need to introduce you to Jim. We really need someone to do testing on cardiotoxicity for this drug we're working to develop. And I said, yeah, sure, give me your card. And I've had many interactions like that over the last 10 years. And most of the time, nothing ever happens with it. But I knew Laura. I just got this sense from meeting her that she was going to contact me. And it was like, I think, a week. She'd emailed me. She came down on her way back from visiting her parents in McAllen and visited my lab. It was shortly after that she introduced me to Jim and the team from North Texas, and we kind of took off from there. So I really want to thank Laura and the Rutledge Cancer Foundation, obviously, for the support of this work. And I'm really invested in this. Our lab's got my first federal grant actually came using this, which is a big milestone. I was pretty happy with that. So it's an opportunity that I got that was just one of those things that happens in your career that wasn't planning on it, but I think it's turned out to be way better than I ever expected. And I want to thank Cana Therapeutics, because Jim has been so wonderful to work with. And I've learned so much from him too, so it's great learning kind of some things about the industry side of this and continuing to think about where we're going to take this. And then my lab, I have a great team. I have Logan Davis, Catherine Crouch, who did most of that work that I showed you. And then Jessica Jarnigan's a MD PhD student that's in my lab and hopefully going to come back in 18 months and hit the ground running. And then two collaborators that are key for this project are Alex Pertzum-Leadis, who I work with in San Antonio, and Jason Eustine, who is a pediatric oncologist at Texas Children's and a sarcoma specialist who has helped really give this project credibility from the sarcoma side, and also has some good models that we're able to draw upon. So I'm going to end there. If you have any questions for me, come find me. I'm happy to answer them.